So our last video was about the proposal of the model of the DNA, the double helix DNA, and this model was proposed by Watson and Crick, and this model was based upon the experimental findings of other experimenters such as Chargaff, Rosalind Franklin and Wilkins. And not only did they came up with this brilliant idea by analyzing the data that they had at their hands, not only did they propose the structure or the model of the DNA, they also proposed something that the world was not ready for, which was the mode by which DNA replicated, made copies of itself. And that particular mode which they proposed was known as the semi-conservative mode of replication. Now, in our previous video, we had seen the three different modes that were popular, the three models that were popular at that point of time: the semi-conservative mode of replication, the dispersive mode of replication, and the conservative mode of replication. Now, these guys, the work of these two guys invoked interest in the scientific fraternity around the world, and two such individuals who were intrigued by their proposals, by their idea that they had put forth, the idea of the model of the DNA and the semi-conservative mode of replication in particular, was received very positively by Messelson and Stahl. Now, when I say positively, I don't mean that they accepted it, but rather what they did was they were like, all right, let's get together and start working on experimentally determining what is the mode of replication of DNA in organisms. So these two gentlemen came together and they designed a wonderful experiment, and there's actually a movie on it on YouTube. It's called The Most Beautiful Experiment by Messelson and Stahl. And anyways, let's not talk about it. I'm not here to promote a movie, but let us just see the design of their experiment here. So in their particular experiment, they made use of many things, but two important contents or components that they made use of was the E. coli bacteria. The bacteria E. coli, one of the coliforms that we've studied in our previous classes. And also they used a heavier isotope of nitrogen. Now, nitrogen usually that we find in our environment, in our surrounding, in the variety of salts, nitrogen containing salts, is nitrogen 14. This is one of the most abundant isotopes that we see around in our world. Now, this nitrogen is also a part of proteins in the living systems and of the DNA in the living systems, and even of RNA. Now, in proteins, nitrogen is incorporated in amino acids, and in DNA, nitrogen is incorporated in nitrogenous bases, the 4 nitrogenous bases that we have discussed: the adenine, guanine, thymine, and cytosine. Now, they made use of this N15 isotope. And how did they make use of? We'll just see it in a little while. Now, these were the two major components of their experiment. They used wide variety of other things, but these were the two major ones, because these are the two components that we are going to talk about. These are the two components that we are going to understand how they inferred their experiment, etc., etc. Also, they devised a very wonderful technique of separating DNA molecules, of basically separating out or purifying, not exactly purifying, but analyzing DNA molecules, and that particular technique is known as the density gradient technique. Now, I'll touch upon this particular technique, and also will give you a separate video on this particular technique, so that if there is any particular problem in understanding right here, you will get a very clear picture from that particular video. Anyway, so we'll also study about the density gradient technique, because they made use of it, particular technique, in analyzing the results of their experiment. Now, there were these 4 major steps that they made use of in their experiment. The first step was to culture, and we'll just get into it, what culturing is. So the first step was to culture the E. coli. The second step was the isolation of DNA and performing the density gradient centrifugation. The third step was subculturing in normal nitrogen containing media of E. coli, and the fourth step was DNA isolation again, and density gradient centrifugation, and then repetition of step number 3 and 4 for a couple of times. And now this is going to become a little confusing if I don't get into the details of each of the steps. So let us look at the first step. So the first step that these two individuals did is that they prepared a culture media. Now, when we say a culture media, we mean to say food for the microorganism. So they in a conical flask probably, or in some other container, they they put a particular nutrient medium, they put food for the bacteria. Now nutrient media is not raw material. For example, it's not like potato or meat that we put in on which we expect the microorganisms to grow. Yes, they will grow, but there is a wide variety of flora, microflora, that will start growing up. So these culture media are basically nutrients that are specifically designed for a microorganism or a type of microorganism. So they first of all devised a culture media for E. coli, our organism for the experiment. They devised this particular media. Now this particular media is going to provide macronutrients to this particular bacteria and also will provide micronutrients to this bacteria. Now major macronutrients it would provide is carbon, hydrogen, oxygen and nitrogen. Now they were more interested in nitrogen because nitrogen is a part of the nitrogenous bases in the DNA. So what these guys did, whatever material they were using as a source of nitrogen for the microorganism, they modified that particular source. Let us take, I'm not too sure what they made use of. They probably made use of a salt somewhat like ammonium chloride. So ammonium chloride is a salt which basically is used in wide variety of bacterial cultures to supply nitrogen, probably a bit of chloride ion or maybe hydrogens, but primarily nitrogen. What they did was they obtained a modified salt which was a source of nitrogen. Let us assume for our purpose they probably used ammonium chloride. Now the normal regular ammonium chloride that we find in the labs or in the market usually has nitrogen 14 in it. They obtained a modified salt, and this particular ammonium chloride, in this the nitrogen had the atomic mass 15, and this was again NH4Cl. Otherwise this particular salt was exactly same to the normal ammonium chloride. The only difference being that this was the heavier isotope. It had the heavier isotope of nitrogen. So they prepared culture media containing a heavier isotope of nitrogen, and that heavier isotope of nitrogen, if the bacteria grew on it, would be taken up by the bacteria. Now, when these E. coli microorganisms were grown over and over on this media, so let us say we had a starter culture of E. coli, we added up into this particular thing. So one generation it multiplied, 2, 3, 4, 5, let's say we had like 5, 10, 15, 20 generations in this flask. We take out all of that bacteria, we transfer it into another flask, another conical flask containing again heavier isotope of nitrogen. Let's put it this way, let's put it N15 here. We again, that particular media also had this heavier isotope, and then we cultured it once again and many a times. Why were we culturing this E. coli bacteria over and over in a culture media that contained heavier isotope of nitrogen 15? We were doing this, or sorry, those guys were doing it. We were not doing it. They were doing it so that all the E. coli microorganisms or these particular bacteria definitely incorporated into their DNA this heavier isotope of nitrogen. Let's use a red marker for this. So now their DNA was rich in nitrogen. Obviously it had to be rich in nitrogen, but the speciality was that the DNA now contained nitrogen, heavier isotope of nitrogen, which is nitrogen 15. Also, undoubtedly, the protein content was also rich in nitrogen 15, but we are not bothered about it. We don't, anyways, these guys didn't want to use the proteins for their analysis, so they were not bothered about it. The important thing was that now the DNA of this particular E. coli that was under experiment, the DNA was labeled with a heavier isotope of nitrogen. Now they grew it for generations and generations, and then they obtained something called as a stock culture. Probably they got something called as a stock culture. Now stock culture is basically a kind of a storage, a large storage of these particular bacteria. So let us assume that they had this. Let's say this is the stock culture. This is the stock culture of E. coli with nitrogen 15 in its DNA. Now this was the first step, culture. I hope this part is clear to you. Now they proceeded to the second step, and the second step was the isolation of DNA. Now they took a certain amount from the stock solution. Maybe this was, let us assume, let's say this was 10 liters of the stock. I'm not too sure what the experimental details exactly are, but let us assume that they probably had, let's say, 10 liters of the stock. So they took out a certain amount. Let's say they took out, let's say, 500 ml of this particular bacteria, and then they expose this particular E. coli bacteria, zillions and zillions of them, to certain chemical treatment or physical treatment in order to break down the bacillus. So we have the E. coli, they would have broken the cell wall, the cell membrane, and then this particular process would have taken place inside a test tube or a conical flask. And then after breaking down all of these E. coli from the stock solution, they would have got cytoplasmic content, they would have got a large number of micromolecules, macromolecules, proteins, etc. And one of the components would have been the DNA. And now what they did was they took out all the other contents using certain technique and purified and got this DNA out. So the second step was isolation of this DNA, the isotope labeled DNA. Now all the DNA that they collected was rich in nitrogen 15. Right, so this DNA was different from the DNA of any other normal E. coli, because the DNA of a normal E. coli, naturally occurring E. coli, will have the normal isotope of nitrogen in their DNA. Now the next step that they did was that they exposed this DNA to a technique called the density gradient centrifugation. Now let us just proceed towards this technique called the density gradient centrifugation. I'll need a new page and let me describe to you what is density. Let's write it here. Density gradient centrifugation. Now I hope everybody knows what centrifugation is by now. Centrifugation is a process or basically a analytical process in which we expose solutions to centrifugal forces by rotating them in a particular machine. So we rotate them at very high speeds, the solutions, and the solutions, which are usually in a tube, in a separating tube like this. So let's say if we have a solution like this, so if you expose this to centrifugal force, which is an outward acting force, the force will act outwards or downwards. I'm representing it because if the tube is rotated, right, if it is spun around, let's say we have a tube here, it will spin out like this. So we'll have tubes like this. So the force that they experience is outwards, and this is the outward force. So when a particular solution experiences a centrifugal force, the heavier or more massive particles, so heavier, let's use these terms here, heavier or more massive particles or more dense particles will be affected more by the centrifugal force, and those particles will start to move to the bottom. The densest and the heaviest or the most massive particles will go to the bottom, the less dense will form a layer on the top, and the least dense will stay on to the top, right? So this is called centrifugal. This process is called centrifugation, and this is a way of separating molecules of different densities. So I hope now you understand what is centrifugation. Now what is density gradient centrifugation? What these two individuals actually did was really amazing. Something that they thought about was really amazing. What they did is that they, in a tube, in a centrifuge tube, they took out, centrifuge tube is a specially designed tube in which centrifugation takes place. So they took out a centrifuge tube, and inside that tube they took water, and to that water they added a salt called cesium chloride. Now cesium chloride is basically a salt in which cesium, it's a very heavy element. Now it has an atomic mass somewhere, I'm not too sure, probably 140 AMUs, but this is a very heavy atom. Cesium atoms are extremely heavy. Now, and let me just rub this part away because I'm not too sure. What I'm sure of is that this is a very heavy atom. It's beyond 100 atomic mass units. So it's a very heavy atom, and they deliberately use this. Now when cesium chloride is added to water, it dissociates to give cesium positive ions and chloride negative ions. And water anyways is a polar molecule. So when I say polar molecule, I mean to say that there is a distribution of charges. So oxygen is slightly negative and hydrogen is slightly positively charged. So the two atoms are slightly positive charged, oxygen is slightly negative. And due to the presence of water and these ions inside, there is this electrostatic interaction between these two species. So the cesium atom is basically, it interacts, it is attracted towards the negatively charged oxygen atom, and chloride atoms are attracted to the hydrogen atoms. So you see in this whole particular solution, the cesium molecule, the cesium ions will be attracted to water molecules. And let's say, let me also draw another water molecule here. So let us say we got the positive charge and the negative charge, and there are these bonds, and so cesium is also attracted. So you see, we had such a mixture in here, right? So we had a solution of water and cesium chloride, in which cesium was interacting, was forming weak bonds with negatively charged oxygen, and chloride ion was also forming, was basically electrostatically attracted towards the hydrogen atom of the water molecule. And so was the condition, and it was a uniform solution to start with. It was a homogeneous solution, water and cesium chloride. Now what these guys did was that they exposed this tube containing water and cesium chloride to very high centrifugal forces. They put it into a machine called the okay, I hope I'm audible. They put this particular tube containing water and cesium chloride into a centrifuge called the ultracentrifuge. Now ultracentrifuge has the ability to basically rotate at extremely high speeds. They can rotate at 100,000 revolutions per minute. So they can generate extremely high centrifugal forces. What they did was they exposed this particular solution to 50,000 RPMs, 50,000 rotations per minute. And while this solution was rotating around in the centrifuge, while it was going around, the massive centrifugal forces came into being, they came into action. And what they did was, now out of all of these atoms, if you look at hydrogen, it is the lightest out there, the least massive atom. And then we have got oxygen, and then we got chloride, the most massive being cesium. Now due to this downward acting or outward acting centrifugal force, let's just use a smaller eraser and let us also reconstruct our centrifuge tube. Now due to this downward acting centrifugal force, the cesium atoms, which were uniformly distributed around, started moving downwards. And what we had was, we had a large concentration of cesium atoms at the bottom, right, to start with. Now remember that this process of ultracentrifugation was carried out for hours to days, many hours to a couple of days. This process was carried out. And what was happening, the more massive cesium atoms started moving down. The chloride and hydrogen and oxygen atoms, water molecules, they were not affected greatly, but certainly because there is a centrifugal force, such an enormous force, so they were moving down, but they were also coming up, because you cannot condense things by bringing the atoms together. We cannot produce such an immense force. But anyway, cesium atoms were started to coming down, but they could not form a pellet. They could not collect together. Now any guesses why? Now the reason being, cesium atoms are positively charged, and because all of them were positive, they were also repelling each other, so they could not form a pellet. And also, as their concentration was increasing in a particular place, even under the effect of centrifugal forces, diffusion was also acting. Diffusion was also evident. So cesium, because it was concentrated to the bottom, some amount of cesium was still moving outwards, right? And there was also this repulsive force. So as you can see here, there was a certain kind of a gradient or a difference, a gradual difference in concentration. So gradient is a difference in concentration. So a gradient was set up in this particular tube. Why? Because heavier cesium atoms first of all moved down, but because all of them were positively charged, they could not stay together. But most of them had to stick to each other because of centrifugal force, but some of them had to escape because of the positive charge repulsion, right? Light charges repel each other. The other phenomena because of which all the cesium ions could not accumulate at the bottom was the process of diffusion. When the amount of cesium ions at the bottom or at this particular lower end of the tube became too high, they started diffusing to the areas where they were low in concentration. And when this process was carried out for a couple of days, this entire state came into a state of equilibrium. It achieved a certain equilibrium state, by which we mean the bottom of the tube actually had the highest gradient or highest amount of cesium ions. And then as we moved up, the concentration or the amount of cesium ions became low and low and low, and at the top we had the least amount of or concentration of the cesium ions, and we had a gradient. And now we can understand this gradient in terms of, so far we've understood the gradient in terms of amount of cesium ions, but there is another way to look at it. Now if you see, let us say we divide this tube into parts of equal volume. Let's say from here, let's use a different marker. Let's say let us divide this 1 and 2 and 3 and 4, and let's say this is the final 5. Let's assume that all of these are equal divisions. So let us say let's assume that this is 1 ml. This is 1 ml volume, right? And this is, or let's say this is 1 cubic centimeter of volume, and so is this, and so is this, and so is this. So this is the same volume basically. But if you look at this particular volume here, it has the least amount of cesium ions. And if you look at the last column here, it has the most amount of cesium ions in the same volume. So at the bottom, 1 cubic centimeter had the highest amount of cesium ions. What does that mean? What does that mean? That means that the density of cesium at the bottom of the centrifuge tube was the highest. So the density at the bottom was the highest, and the density of cesium was least at the top. So now this is what was achieved at the end of this particular process. Density gradient centrifugation allowed them to generate a solution in which there were layers of different densities, right? So now what next? Let us go back. So I hope everybody's understood what density gradient centrifugation is. And now what was the third step? Now, okay, now at this particular moment, we won't go into the third step. We'll still continue with the second step. What they did next was the DNA that they had isolated, this nitrogen 15 that they had isolated, right? I hope you remember. From the stock solution, they took out this particular bacteria, now in which all of these E. coli bacteria had nitrogen 15 in their DNA, right? Now they had isolated this DNA, purified this DNA. Now they are ready with this DNA. What they did is they took the centrifuge tube, okay? They took the centrifuge tube, they added cesium chloride solution. They added, I'm writing aqueous, so this is cesium chloride solution. They added cesium chloride solution. To this solution, they added a predetermined fixed amount of this DNA. So they also added DNA to it. So let us put DNA right here in blue. So let us say this is the DNA. Now, will the DNA stay on the top, or will it sink down without centrifugation, I mean? Now, if there is no centrifugal force acting, the DNA has a certain density. It will in this particular solution, because before centrifugation, the entire column of cesium chloride has a uniform density, has the same density, uniform density. So the DNA, if it has less density than cesium chloride, then it will experience an upward buoyant force and it will float on the top, right? But if it is more dense, then it will start to sink. But they didn't wait for it. What these guys did, they added the DNA, and here we had the cesium chloride solution. Let's put the cesium chloride solution. There was no density gradient set up at the moment because centrifugation had not started. But now what they did was they put this tube with the DNA with cesium chloride inside the ultracentrifuge, and they started spinning it. And they started spinning it. They started generating that high amount of force. And this experiment again continued for a couple of hours, as we discussed, for a couple of hours, probably a couple of days. One of the textbook that I read said it ran for a couple of hours. The other book says it ran for like 2 to 3 days. This experiment was run. Anyway, so the key point being, now the DNA is here and we have this tube. Now inside the tube, let us magnify it a little. Inside this particular tube, at the end of the session, so let us make this is the tube. At the end, when the equilibrium is achieved, we have regions of different densities. So we have the most dense zone at the bottom and the least dense zone at the bottom, at the top. So we have the least density here, least dense zones here, and we have the highest density at the bottom. And now this nitrogen 15 started to sink. Now, how long will it sink? See, as this DNA is experiencing a centrifugal force, it will move down, right? It will move down into the solution. But then it will reach a certain point where, let us say this is our DNA, and let us say the entire DNA, which is rich in nitrogen 15, has reached this particular point. Now, just below this point, the density of cesium chloride is higher than the density of DNA. So it cannot sink any further because it is going to experience an upward force. And this band of DNA, the entire collected DNA, which is rich in nitrogen 15, cannot also move up. Why? Because the density above it is less. So basically this DNA, after a couple of hours of centrifugation or maybe a couple of days of centrifugation, this entire DNA finds itself fixed in a particular position. Why? Because it has found its sweet spot of density. That is the spot where its density matches the density of the cesium chloride graded solution inside. Right? I hope you understood this. So they performed this step with the first step. They did the culture, they prepared a stock solution, they took this nitrogen 15 rich DNA out, purified it and exposed it to density gradient centrifugation, and then ran it for a couple of days, and then they get this band. And this is the band of DNA which completely contains nitrogen 15. It has no nitrogen, which is rich in nitrogen 14. And now let us look here. So this is called the day 0. This particular sample of DNA is called the sample of day 0, not the day 1. We'll see why. So here, let us have a look at this, and I'm going to use a pointer now. So this is the tube, the ultracentrifuge tube, and at some place here they find their first band. And how much of DNA was there? 100% of the DNA was collected here, because the entire DNA is rich in only nitrogen 15. So here they got their first band on day 0. Now what these guys did in the next step was, I hope this is clear to you, and in the next step what these guys did, let's use a larger thing, and okay, let's not destroy this tube here, but let's just take these things out, okay, because I'll need to draw it again. So just give me a little time so that I can actually draw the centrifuge, the destroyed, damaged centrifuge tube again. So here is the centrifuge tube once again. So now what these guys did, after performing that experiment on, and so let's say this was like day 0, this experiment was going on, right? And they will get their result of day 0 after like whatever time the centrifugation takes place. Now at day 0, again, what they do was, from the stock solution, if you remember, from the stock solution, they take a little amount of E. coli. From the stock solution, they take a little amount of E. coli. Now, all of that E. coli, what they are taking from the stock, its DNA is rich in nitrogen 15. There is no nitrogen 14. And what these guys do, they transfer a little bit of this E. coli into a new culture media. So now they have a new culture media. Okay, so let us draw another conical flask. And this is where the entire catch of the story is. So this is, so they take another conical flask, they plug it with cotton, but they'll certainly have to remove the cotton later on to add the bacteria, and they make a new nutrient media, fresh nutrient media, again the same kind of nutrient media. But now this nutrient media is different. It does not contain nitrogen 15 isotope. The source of nitrogen, let us assume again ammonium chloride that they use this time, is the normal ammonium chloride, by which I mean it contains nitrogen 14. It does not contain nitrogen 15. So what is going to happen? So anyways, what they do is they take the stock E. coli, let us say they take a couple of certain number of E. coli, and all of those E. coli have nitrogen 15. They have the heavier DNA inside, and they put it inside here, right? And how long they incubate it? Let's say for around 20 minutes, which is the time taken by the bacteria to double up. Okay, so what they do is they only allow this E. coli to simply just double up, which means they only allow this bacteria to multiply once, and what will happen there is something that we'll see in our next slide. Let's just have a look at this here. Okay, let's not look at this here because okay, let's rub this off and make some space for ourselves, and let's see what happens. So here I'm going to use a red marker to show the heavier DNA. So let us say we had this heavier DNA, right? We have the heavier DNA here, and this contains nitrogen 15. Now when this, and this is from the E. coli that was grown over many generations in a nutrient medium containing heavier isotope of nitrogen 15. Now a little bit of this has been taken and put into a normal nutrient media. So what they do is they take a bit of these bacteria containing the heavier isotope in their DNA and they place into the normal media. And now what will happen, and how long do they keep this bacteria there in that particular media? They keep it there long enough only for one cycle of multiplication. So assuming that we had one bacteria inside, they only allowed this to happen, right? So what will happen? If this particular E. coli, certainly I've made a sphere, it's not E. coli, but let us just assume that this is the bacteria E. coli, it had to be a bacillus. So let us say this had nitrogen 15 in it, and now they are exposed into a nutrient media in which the source of nitrogen is normal. So what will happen is that they will make another copy of their own cells, and half of the DNA, we are just assuming, because now half the amount will remain the same, but as two copies have to be made, half of the DNA will contain nitrogen 14. Why is it so? Because see, the original strands, the bases in the original strand, nitrogenous bases in the original strand had nitrogen 15. But if they have to make the other half, if they have to double up, if these strands have to double up, they will have to use nitrogen 14. And if they use nitrogen 14, what do we get? We get half of the nitrogen 14, half of the DNA will contain nitrogen 14. So they were not sure whether this will happen, because this is what I am showing is conservative, is basically the semi-conservative mode of replication. Probably something else was also happening, but what definitely would have happened is 50% of the DNA after the first cycle of division, 50% of the DNA would contain nitrogen 15, the heavier isotope, and the remaining 50% of the DNA would be rich in nitrogen 14 isotope, right? So this is for sure that would have happened. So now what they did, they allowed these organisms to grow for, let us say, 20 minutes or whatever amount of time it took this particular E. coli bacteria to double up. And now exactly after that period of time, they took all the E. coli out, the doubled up E. coli. I hope you're getting this idea. So when they took all of that E. coli out, they again broke the E. coli. Let's go back to the steps. So the third step, now we're in the third step, subculture, right? You remember we have done the subculturing. We've taken the stock, a little bit from the stock solution and placed it into a medium containing the lighter isotope of nitrogen. So this is called subculturing, right? So we've done the subculturing and we have allowed the bacteria to just multiply once. And now what we are going to do is we are going to isolate its DNA. So what these two guys did, after 20 minutes from the first generation of the bacteria that was grown on the normal media, they took those bacteria, they broke them up, they isolated their DNA. They had no idea what kind of DNA would they get. Would they get a completely heavier DNA or would they get a lighter DNA? What kind of DNA would they get? And they took this DNA and they exposed this DNA again to the density gradient technique. They placed this DNA, let's just use that particular slide. So they took another centrifuge tube, they added cesium chloride to it. So we have cesium chloride aqueous solution, and then they added this DNA on the top, and they started this process of density gradient centrifugation for the same amount of time that the first experiment ran for. You remember the first experiment where we got the band for the heavier DNA, the DNA that contained heavier isotope of nitrogen, right? So all the other experimental conditions remained the same. And after they had stopped, after they had finished, right? Now this DNA will experience centrifugal force and it will go down, it will settle down somewhere, right? It will settle down somewhere. And what these guys found astounded them. And this is what they found. And here it is. The first generation. Now this is called day 1. I hope you now remember day 1. Day 1 is basically the first generation of bacteria that was grown on the normal media with nitrogen 14. The band of DNA had shifted slightly above the original band. So the original band of the heavier nitrogen 15 was here, right? And this was basically the 0 generation, or let's say day 0. And the band of the first generation is now a little up, which means the density of the DNA has reduced. The density of the DNA had reduced. Now, also at the same time, what they had done, right? I just skipped my mind, but anyways, it's not too late. So we know where the band of nitrogen 15 is found. It is found here. Likewise, these two gentlemen had also performed density gradient centrifugation for DNA of E. coli, which contained the normal N14 isotope. They had already performed it. It skipped my mind, but let us just do it now. They had already performed an experiment for this, and they knew that the band for the DNA that contains the normal isotope of nitrogen, they would find this particular band. Let's use a different marker. Let's use yellow. So nitrogen 14, they would find a band here. They had already conducted these two experiments. They were easy to do, right? Nitrogen 15 settled here, the DNA containing nitrogen 15 settled here, and the band for nitrogen 14 was here. So they already had these two informations. And now what they found of the day 1 generation, or the culture media that was for the first time transferred into the normal media, that was allowed to multiply only once, for that particular DNA, they found the band exactly in the middle of the two, right? So this was the band for nitrogen 15. This was the band for nitrogen 15. They already had this nitrogen 14 being lighter will stay on the top. Nitrogen 15 was down. But at the end of the first subculturing here, you see, we took something from the stock and transferred it into the normal media. After centrifugation, the band that these guys observed was found exactly in the middle. Not in the same tube, obviously, but in a different tube. Right, so let's go back here and try to understand. So they already knew about this. This is nitrogen 15 band. They already knew about this. This is the nitrogen 14 band here. So it is somewhere here. So this is nitrogen 14 and this is the band for nitrogen 15. But for the first generation, they found the first band somewhere here. It was exactly in between the two. Now if it was in between nitrogen 14 and nitrogen 15, this meant its density was exactly in the middle of the two. Right? That was the first generation. Anyways, let's proceed further and let us see what happens next. Now what these guys did, and this is what we are going to do. So this is the first generation. And now what they did was they took these particular bacteria again. Okay, they didn't use all of them up. Okay, I hope you can realize what I'm talking about here is, let us say this is the day 1 bacteria. These are the bacteria from day 1. So they didn't use all of those bacteria for density to obtain DNA. They also retained a couple of them. They retained a couple of those bacteria from day 1. And what they did with those particular bacteria is they cultured them again on a normal media. So they took a normal culture media. Oh, I'm drawing a petri plate. Let us just stick to our conical flask. So let us say they have this particular conical flask, and we'll call it like the day 2. Right? So this is going to be a very big video, but I think we should not be bothered much about it. So this is basically day 2, and in day 2 what these guys did was, see, from the first day, from the first normal cycle of multiplication, single multiplication, they used those particular bacteria to obtain the DNA, but they retained a few from there. Whatever had multiplied, after 20 minutes they took some of them from there, and this is, we can assume this is the generation of day 1, and then they transferred it into culture media once again. What kind of a culture media? Culture media that had nitrogen 14. They transferred this here and allowed it to multiply only once, just like in the previous experiment. They just allowed it to multiply once. So what happens, this particular bacteria, it divides in a certain period of time, let's say 20 minutes, and it multiplies and it gives double the amount of thing. And what they find, and then after those 20 minutes, they again stop. They break these bacteria. They keep some of them. They keep some of them for the next step, right? Some of the bacteria from day 2, and they lyse the other. They break down all the others. They isolate their DNA. They purify this particular DNA and expose this DNA to density gradient centrifugation. And once this was exposed to density gradient centrifugation, this is what they got. And this is day 2. This is day 2. And on day 2, they got 2 bands. They didn't get 1 band. There was no band at the density level of nitrogen 15. There was no band. It simply had vanished. But now we had a band between nitrogen 14 and 15. We had a small band here. And then we had an equal sized band above it. How above it? Exactly at the level of nitrogen 14. I hope you're getting this particular point. And then what they did is that they performed this experiment again. The bacteria that they had preserved from day 2, they cultured it again for like 20 minutes so that these particular bacteria doubled up, right? Then they isolated their DNA also. After isolating and purifying their DNA, they exposed it to density gradient centrifugation, just like they've done for the previous thing. The same process was repeated. And after density gradient centrifugation, what was observed, that there was no band observed at the density level of nitrogen 15, but there was still a thinner band, much thinner band that appeared at in between the nitrogen 14 and 15 at the same level, and the band on the top that was observed was the thickest, and it belonged to the density of nitrogen 14. And now they continued this process, but we are not going to go much beyond this particular point. And so what are we observing here? So what we are observing at the end of their experiment, and they ran this experiment multiple number of times with multiple samples of DNA. They repeated this experiment over and over again to confirm that their findings were consistent. So now what they found was the first band was the thickest, and it lied here at this particular level, nitrogen 15. The other band was equally thick, but it lied between the nitrogen 15 and nitrogen 14 band. But then in the second generation, they found 2 bands, and both the bands were equally sized, 50% here, 50% here. And in the third, let's call it the day 3, the band between nitrogen 14 and nitrogen 15 became smaller. It was much lighter, which means we had lesser amount of nitrogen, whatever heavier nitrogen was here, and we had most of the DNA had the density of nitrogen 14. Now, how do we explain this experimental finding? And these gentlemen then went on to basically compare their findings here with the different models, and let us have a look at those particular models. And here we have those models. So let us say whether these findings can be explained by these particular models. So let us have a couple of bands ready. So let us say the parental generation. So let me just draw. Now these are going to be a little rough diagrams. Assume them to be nearly the same. Okay, so assume that these 3 are identical centrifuge tubes that have experienced density gradient centrifugation, and inside of them I'll draw the 3 samples. So where did we find the samples? And let us also have one more tube out here, and I'm going to tell you what this tube is about. So we have like a couple of bands. So let's use a green. So this band on the top is nitrogen 14, right? And let us say with the red down here, we have a band for nitrogen 15, right? So what about the first generation? In the first generation, where was the band? Day 0, the original microorganisms, the heavier strain, right? The parents. So let's go here. Let us just see if semi-conservative mode of replication fits with the finding of the experiment. So the first step of experiment, day 0, what was exposed to density gradient centrifugation? Only the parents. Remember, only the stock solution was taken. Only the stock solution was taken on day 0. And so on day 0, let us say that this is day 0. What do we get at day 0? At day 0, we get this. This is our first band, right? So we have our first band here at the level of nitrogen 15. So first band is nitrogen 15. Now let us look at the DNA, and this is like day 0, and these are representing day 1 and these are representing day 2, or generation 1 or generation 2. Let's not call it day 1 or day 2, we should have called it generation 1 or generation 2. So at generation 1 or day 0, the entire strands were rich in nitrogen 14, you see. So this is the parent strand, right? Here we are only showing one strand, but no matter how many E. coli were there, how much of DNA was there, all the DNA had nitrogen 14. And that's why the DNA that underwent density gradient centrifugation settled at this particular level, the level of nitrogen 15. I hope this is clear. What do we find at day 1? Let us look at day 1. And at day 1, they found a band between nitrogen 14 and nitrogen 15. So let's go. At day 1. So at day 1, basically, so let us understand this. Let us say this is our control. This is our control experiment for the experiment. So what do they find? On day 2, they find a band. Let us use a blue color. They find a band in between. They find a band here. They don't find a band at the bottom, but rather they find a band here. Now, can the presence of this band between nitrogen 14 and nitrogen 15 be explained by semi-conservative mode? Let us see. So the microorganism, the E. coli, was allowed to replicate only once. So if it has to replicate once, the strands separated out, as shown here. You can see the pink strand. The pink strand contains heavier nitrogen according to the semi-conservative mode of replication. We are only trying to understand whether semi-conservative mode of replication fits the experimental outcome. So according to semi-conservative mode of replication, the two strands will separate out and then they will synthesize a new strand. So we have an old strand here, we have an old pink strand here, and the new strand is synthesized, and the new strand contains nitrogen 14 isotope. And because these particular DNA sequences, the newly synthesized DNA sequences, are a mix of nitrogen 15, see, in this particular strand, and a mix of nitrogen 14 isotope, both of them, because it's a combination, the density of these particular strands of DNA that were generated during D1 should actually lie in between nitrogen 14 and 15, and that is what was found. And that is what was actually found. See the experimental, and see the entire DNA. There is no DNA that belongs to nitrogen 15, and there is no DNA that belongs to the gradient or the level of nitrogen 14. Right? That is what they did. And see here also, we have no DNA that is purely made up of nitrogen 15. There is no DNA that is made up of purely of nitrogen 14. So anyways, let us not conclude right away that the semi-conservative model is actually conforming to the experimental findings. Let us look at another generation. So in the next generation, if the semi-conservative mode of replication is true, if that is how the genetic material replicates, then what will happen, that these two strands, so the pink one, I'm going to use a red, the pink one, let's take the first one here, the pink one will separate out, and then the blue one will separate out. The pink one is nitrogen 15, and likewise here also the same thing will happen. We'll have another lighter strand and a heavier strand, and then all four of them will develop their own newer segments, but the newer segments will only be made with nitrogen 14. So we get this, right? Newer strands will contain only nitrogen 14. So what do we have now here? We should now get, let's say 50% of the DNA should belong to nitrogen 14, nitrogen 15 band. And where is this band? This is actually the nitrogen 14 and 15 hybrid zone. This is that zone of the combination of these two densities. So we should get, according to the theory, we should get 50% of this, and we should get a 50% band in what particular region? In the region of nitrogen 14, the density nitrogen 14. Half of the DNA should lie there. And did it actually happen? Let's go back and see. have a look, my dear friends. Here 50% of the DNA actually has only nitrogen 14, and 50% of the DNA lies in that intermediate region of the density nitrogen 14 and 15. Bingo. They were able to actually point out that the semi-conservative mode of replication is actually the mode of replication of DNA. But let's not hurry up way too fast. Let us also evaluate the other two modes of models of replication of DNA proposed by other individuals and see whether do they actually fit in somewhere? Do they fit in according to the findings of Messelson and Stahl? So let us look at this conservative mode. If DNA replicates through the conservative mode, then at the end of first generation, what should we have? Half of the DNA, 50% at day 1. So let's call it still day 1, D0 and D2, okay, for our convenience. So if the conservative model proposed for the replication of DNA is a correct model, then at the end of the first generation at D1, 50% of the DNA should belong to the nitrogen 15 band and 50% of the DNA should fall in the region that belongs to nitrogen 14 band. And let us look at the experimental finding. Is it so? But at day 1 we just have one band. 100% it lies between the two bands. So experimentally this theory of conservative model of replication was knocked out. It was thrown off the stage and it was basically nullified. There was no more legibility to this particular model. What about the dispersive model? What about the dispersive model? At the end of the dispersive model, what is going to happen? See, so D0, again we have only one band. It proposes. So let's go back and see. Day 1 we only have one band that falls in nitrogen 15. Okay, fine so far. In the first generation, you see now it will break from here, it will break from here, it will probably break in between here, and then it will generate newer strands. So what is going to happen? Let us just assume that during the first generation, 100, so let us say 50% of nitrogen was heavier and 50% was lighter in this first strand, and so was the case here. So let us say in the first generation we will only get one band after density gradient centrifugation, and that band would belong to nitrogen 14-15 zone. And actually it does, right? Actually the day 1 band is the intermediate band, so we cannot right away discard it. But let us look at the next generation. What is going to happen in the next generation? In the next generation, you see all the strands. There are these different strands, and all these strands have some traces of nitrogen 15 in them, which means none of the DNA, no matter what particular DNA we have, none of the DNA will fall in the region of nitrogen 14 in the density gradient. And this is day 2 that we are talking about. And let us look at day 2. Day 2, according to the experiment, we already have half of the DNA band in the zone of nitrogen 14. But according to the dispersive model, we just cannot have that band because each DNA strand has nitrogen 15 on it. And if this model were to be true, we shouldn't have got this band at all. And so this amazing experiment by Messelson and Stahl eventually proved that the semi-conservative mode of replication was actually the mode of replication as proposed by Watson and Crick. Now this is an awfully long video. I hope you stick to it. Thank you.